We have learned that many cellular functions are mediated by proteins that interact with each other. A method to identify genes encoding proteins that interact with a known protein is the Yeast Two-Hybrid System created by Stanley Fields and colleagues. The Yeast Two-Hybrid System is based on understanding the transcription of galactose metabolizing genes. The GAL4 gene regulates galactose metabolism in yeast. The GAL4 protein has two domains. One domain, the binding domain or BD, attaches to a regulatory sequence in the DNA upstream of the genes it regulates. The other domain, the activation domain or AD, activates RNA polymerase to transcribe those genes. Both domains are part of the same gene but can be separated and retain their activities through recombinant DNA manipulation. The GAL4 protein regulates the GAL1 and GAL10 genes. Between these two genes is a promoter element called the upstream activator sequence G, UASG. The GAL4 protein forms a dimer and the binding domain attaches to the UASG. Then the activation domain binds to RNA polymerase, initiating the transcription of the two genes in opposite directions. The transcription of one gene is illustrated here. In the two-hybrid system, two types of yeast expression plasmids are constructed. One type contains the sequence for the GAL4 binding domain fused to the coding sequence for a known protein X. To create this plasmid, the binding domain sequence of the GAL4 gene is cut out and inserted into a plasmid next to a promoter. The bait gene X is then inserted adjacent to the binding domain fragment. This plasmid also includes a selectable marker for yeast, such as URA3. The second type of plasmid contains the sequence for the GAL4 activation domain inserted next to the promoter, similarly to the first plasmid. Then, a library of cDNA fragments potentially containing the genes for protein Y that interacts with X is added to the plasmid. This results in a family of recombinant plasmids, each containing a different cDNA. This plasmid also includes a different selectable marker than the binding domain plasmid, such as TRP1. The BD plasmid and the AD plasmid library are transformed into a yeast strain with mutant URA3 and TRP1 genes genetically engineered to carry a reporter gene. The reporter gene is the LAGZ gene for beta-galactosidase from E. coli, with its promoter replaced by the yeast UASG promoter. The transformed cells are plated on a medium lacking uracil and tryptophan and containing the chemical X-gal. Transformants that grow into colonies on this medium each contain the BD plasmid and one of the AD plasmids from the library. Colonies that can produce beta-galactosidase will turn blue in the presence of X-gal, while those cannot remain white. How is a blue colony produced? The plasmid with GAL4-BD and the bait gene X produces a fusion protein that binds to the UASG promoter of the LAGZ reporter gene. If the plasmid with the GAL4-AD carries a cDNA fragment for a protein Y that interacts with the X protein, the BD and AD fusion proteins will bind together through the XY interaction. This hybrid protein contains both a functional BD and AD, leading to the transcription of the LAGZ gene. The beta-galactosidase produced converts the X-gal into blue color, turning the colony blue. In white colonies, the Y cDNA fragments on the AD plasmid does not code for a protein that interacts with the bait protein X. Therefore, even though the BD can attach to UASG, the AD cannot bind, as the Y protein does not interact with X. Without the pairing of the two fusion proteins, no beta-galactosidase is produced and the colonies Do not turn blue. The Yeast Two-Hybrid System has proven useful for defining protein-protein interaction across various organisms.